Hello and welcome. This is the guided drawing video for 9.1 sexual reproduction in flowering plants. Now, being said, it is a guided drawing, so as I draw, I hope that you would follow along. Please pick up a pen and pencil or any surface that you would like to draw your drawing on. As I go, I'm going to explain a little bit and also label, and I hope that I am going at a pace that you can follow. So if you're ready, let's begin. So as you know, we are talking about flowering plants. So before we go into the nitty-gritty bits, let us first learn about the anatomy of a flower. A flower, as you know, is the reproductive organ of the flowering plants. And there are a few components or a few floral organs, but once you have an idea of how they are laid out, it's quite simple to remember them. So let's begin. I'm using a colored pen for the structure. And we are going to begin with the stigma, which is the part on the flower where the pollen grains will land on. So that's the stigma, followed by a style, which just goes straight down. And then you will draw the ovary. The ovary is basically something big or round or oval shaped. And make sure you draw it a little bit bigger on your paper so that you can draw the structures that are inside. Now in my ovary, I'm going to draw the space where the embryo sac will be. I have this little bit that's hanging or sticking out so that it can support the embryo sac inside. There we go. And inside of the embryo sac, we're going to have the 3 antipodal cells at the top, the polar nuclei in the middle, the egg cell near the micropyle, and the 2 synergid cells next to the egg cell. And voila! That is the reproductive organ, or the female part of the reproductive organ of the plant. Before I continue with the male part, let me just zoom out a little bit, and I'm going to draw the structure that will support the entire flower. So I have the sepal here, which are like the green parts that protects the flower before the flower blooms, the receptacle, and the stalk. And from there, I'm going to draw the anther and the filament, which are the male parts of the flower. So the anther is kind of just like 2 ovals, one in front of the other. And I draw the filament is something that just supports it so that it kind of stands further away from the stalk. Okay, so that's one anther and filament. I draw another anther and filament on the right side. And draw it like that. Oops. It's a bit tricky with technology. You can draw your filament a little bit thinner and your anther a little bit bigger. So that's completely up to you as long as you have the structures in place. There's one more thing, the most distinguishing part of the plant, which is the flower petals. So I'm going to draw the petal here. So you can be creative with the petal part, draw as many or as little as you'd like, and you can even color them in if you want to. Here I'm drawing 4 flower petals and making sure that they look neat enough. There we go. Now it's not the most artistic flower, but it will do for this exercise. Now let's get to the label. To label, I'm going to start with the female part of the flower first. So over here, I'm taking my black pen and we are going to start by labeling the most top part, which is the stigma. So S-T-I-G-M-A, stigma. This is where the pollen grains will land and then germinate. Because on the stigma, you will actually find a sugary solution that simulates the pollen grains to germinate. Right after that is the style. So let's go ahead and write style. And below the style, that is where we have our ovary. So you can label it like this and write ovary, or you could also do it like this, label this whole thing as an ovary. And then this stigma, style, and ovary make up the female organ of the flower, which is called a carpel. So I'm going to write here carpel, C-A-R-P-E-L, carpel. Now let us label the male part of the flower. So as you know, it only consists of two parts, and that is the anther and the filament. The anther and the filament will make up the male organs of the flower called the stamen. Let us also label the other parts of this flower, although they do not produce any gametes. This is the petal, the sepal, and the receptacle. And there you go. Just another tidbit of information. On flowers, sometimes a flower would have only carpel. Some flowers will have only the stamen, but some flowers will have both carpel and stamen on it. It depends on the species of the plant and also the gender of the plant itself. Okay, so that is it. That is the anatomy of a flower. Now, in the next segment, we are going to be learning about what goes on inside of the ovary and inside of the anther. I think it's good for us to start with the male part this time. Before we continue with the lesson, for those of you who are drawing on a piece of paper, I would like you to take your piece of paper, fold it in half, unfold it, and now use the left side for the male gametes and the right side for the female gametes. So you can do a side-by-side comparison between the male and the female. Okay, for those of you who are using this digitally, then do with it however you want. We are going to continue with the development of the pollen grain and the formation of the male gametes. So before we continue, I would like you to first imagine the anther. Okay, so once again, the male part of the flower, and cut it in the middle. So if you're cutting it in the middle, you are doing what is called a cross section, an anther cross section. And the cross section of the anther would look something like this. Okay, so draw along with me for this part. It's okay if your drawing is imperfect. It has 4 parts and 4 microsporangium. So this is what is called a microsporangium. It also has another name. It can be called a pollen sac. So if you see the word sac, H-A-C, it means it is a type of bag and it contains something. So inside it contains numerous microsporocytes. Or what is also called as the pollen mother cell. Okay, so inside are lots and lots and lots of little microsporocytes. Now let us focus on one of those microsporocytes. Okay, so assume that we can take out that microsporocyte. It will look like this. It's a plant cell, so it has a cell wall. And I'm going to label it as a microsporocyte. And this will be a diploid cell. Now this thing will undergo meiosis. So write down here, meiosis. And after meiosis, the cell will be haploid. And it will produce 4 daughter cells. So those 4 daughter cells are now called microspores. We draw them next to each other. They don't look that different from how they were before, but now they are haploid. And I like to put them in the sac together. So this is called a microspore. Actually, if you look here, there are 4 microspores. So it is also called a tetrad. Now let us look at only one of these microspores. Each of these microspores will mature and they are going to become a young pollen grain. Now, a young pollen grain looks pretty much like a normal cell. It is still haploid. Nothing different here. But it will continue to grow and to mature and then undergo mitosis and become this specialized cell called a pollen grain. We're going to start with the exine or the outer layer, the intine, the inner layer, and draw the tube cell and the tube cell nucleus and the generative cell. So you learned this in your slides before. This is now called a male gametophyte. Also called a pollen grain. This thing is the tube cell. This black thing is the tube cell's nucleus or tube nucleus. And this thing is the generative cell. Okay, so this is your male gametophyte, also called your immature pollen grain. Now, this thing is not yet mature. For it to fully mature, it has to undergo pollination. So I would like to write here during pollination. This pollen grain will leave its original anther and land on a different stigma. So the pollen grain lands on stigma. And when it lands on stigma, it germinates. And it germinates. Germinates means it's going to grow its tube cell. So let us have a bit more space to draw. Okay, for this one, I am going to draw the stigma, the style, and the rough shape of the ovary. Okay, we don't need to draw it in detail here. But I do want to draw the pollen grain when it lands. So when the pollen grain lands on the stigma, it's going to germinate and produce the tube. And the generative cell will also divide and produce 2 sperm cells. So here I am going to label. This is the pollen grain. This thing is the pollen tube. The tube nucleus will actually be at the head of the pollen tube. So this is the tube nucleus. And here are the generative cells that have divided by mitosis, and these can be called the sperm cells. And there you have it. Now I'm going to zoom in on this picture in case you are using this on a small gadget and you need a bit more time to look at the picture. So this is when the pollen grain or male gametophyte fully matures. The male gametes fully mature here after pollination. Okay, so once again, before we move on to the next part, when we talk about the development of the pollen grain and the formation of male gamete, we start by looking at the microsporangium, looking at the microsporocyte. In a microsporangium, microsporocyte undergoes meiosis, forms a tetrad of microspores, which are now haploid. The microspores will develop, mature into young pollen grain, and then mitosis into a mature pollen grain or male gametophyte. Inside of the pollen grain, you have the tube cell and the tube cell nucleus and the generative cell. During pollination, it lands on stigma, germinates, fully matures, which is when the generative cell becomes sperm cells, the tube cell germinates and grows the pollen tube. Okay, so this is it for the formation of the male gametes. Now let's move on to the ladies and look at the development of an ovule, embryo sac, and formation of the female gametes. So if we're looking at the entire carpel, we are going to be focusing what is in here. So if we take out this part in the middle, we are going to have the ovule. So the ovule is here, and I'm gonna do the drawing. We start with the part that kind of hangs on to the ovary wall, and we're going to do the integument layer. Okay, so this outer part is followed by another layer in here. Okay, and in the middle is just one big nucleus. Now let us get to the labeling. This entire thing is an ovule. This thing that supports it is called a funicle. It's not as important, but I'll label it for you so you know. This layer here is the megasporangium, or the sac that contains the megasporocyte. This cell in the middle is the megasporocyte. This thing that is on the outside are the integuments. Oh, actually, I'll label it a bit higher. This is the integument. And there is a part of the integument where it is a little bit thinner and the pollen tube will enter through here. So this part is called the micropyle. And there you go. This is the ovary before, or the ovule inside the ovary before we have any meiosis. Now after this, the megasporocyte, which is this big cell with a big nucleus in the middle, it's going to undergo meiosis. So meiosis, here it is 2n. After meiosis, it will become haploid. Okay, let's redraw the whole ovule once again. You're going to be very good at remembering the structure of the ovule after this, because I'm going to have you draw this several times. Okay, so here you have the megasporangium. Once again, it's okay if your drawing isn't perfect. Now, integument still same, megasporangium still same, but on the inside, after meiosis, it will produce 4 daughter cells. But those daughter cells will not be the same shape. One of them will be like pretty big. The other 3 will be pretty small. So we are going to draw them accordingly. So each of these are megaspores. These are megaspores. But as you remember from the lecture, only one of these megaspores will survive. So I'm going to delete here. the biggest one is usually the surviving megaspore. So this one, we're going to write it as surviving megaspore. And this is haploid. These three are also haploid, but they will disintegrate. So these three other megaspores will disintegrate. Okay, disintegrate meaning that they'll just naturally die. Okay, so after this step, the other three will die, and your ovule will once again look like this. I'm just going to draw the ovule very simple. It's not going to be very pretty, and your drawing doesn't have to be pretty either, but make sure that it has the right labels. So now this is surviving megaspore. After the surviving megaspore grows a little bit more, it's going to undergo 3 times mitosis. So mitosis times 3, but with some of them will divide their cytoplasm, but some of them will not. So we're going to draw the product after 3 times mitosis. I'm going to zoom in on the picture a little bit. Drawing the integuments, megasporangium, attach the funicle. I like to start in the middle with the polar nuclei. And then the 3 antipodal cells at the top. One big egg cell at the bottom near the micropyle, and 2 synergid cells that will flank its side. Now we get to the labeling. These are the polar nuclei. These 3 are the antipodal cells. This big cell in the middle is the egg cell, and these 2 are the synergid cells. Now each of these are haploid. So that's it. That's all you will need to do for the female part. So it's a lot simpler as long as you can remember the names. Okay, so let's have a review once again. In the development of an ovule, embryo sac, and formation of female gamete, we start with a megasporangium containing a megasporocyte. After meiosis, it will produce 4 megaspores, but only one of them will survive and the other 3 will disintegrate. You will be left with 1 surviving megaspore that will undergo 3 times mitosis, and the nucleus or the cells that are produced after that 3 times mitosis will migrate to different parts of the embryo sac and produce polar nuclei, egg cell, synergid cells, and antipodal cells. So that is it for the female part. Now that we've learned about how the male gametes and the female gametes are formed, we will now see what happens when they come together in a process called double fertilization that is unique to flowering plants, and it also contributed to their success. So for this segment, I'm going to use several different types of pens. I'm going to use black pen to label, and we're going to draw 3 pictures to show the 3 significant steps in double fertilization. I'm going to draw the stigma and style, but this time I'm making the stigma a little bit bigger and the style a little fatter than before. And we're going to draw the ovary, the funicle, and the integuments. As you see, after a bit of practice, drawing the organs of the flower become a lot more simple. Okay, so the ladies are ready. Let us draw the pollen grain. So during the first, you could say is the first part of the double fertilization process, the pollen grain will land or arrive on the stigma. As it lands, well, it could land because it is being brought over by pollination agents such as wind, water, or insects. And once it lands, it absorbs the moisture on the stigma and it will germinate. When it germinates, what it does, it's basically growing a little tube or pollen tube. Okay, so at this point, the pollen tube will grow and it will move down the style towards the ovary. At the head here, you will have the tube nucleus. This itself is the pollen tube. This is the pollen grain. And something else that is happening in here is that the generative nucleus will divide by mitosis. And it's going to produce 2 sperm cells. So these are called the sperm cells. They could also be labeled by other terms, but in here I'm going to label them as sperm cell. And just so you don't get confused or you don't forget, I would like to label down here as polar nuclei and egg cell. Remember, egg cell is the one in the middle. Okay, so this is the first drawing that you should have on your piece of paper. Now we are going to move on to the second drawing. So for the second drawing in double fertilization, I'm going to once again draw the carpel. So we're going to have the stigma here, the thick style to make it easier for us to draw. Okay, and then the huge ovary. So we're drawing it bigger so that it's easier for you to draw the components later. Okay, over here, the micropyle. Leave the micropyle down there. 3 antipodal cells at the top, polar nuclei in the middle, 1 egg cell waiting right next to the micropyle, and 2 synergid cells that flank it. Okay, so at the top, we still have our germinated pollen grain. And the pollen tube has now grown down. Oops, I didn't draw that very nicely. I'm going to redraw that part. Okay, the pollen tube would have grown down the style and around the ovary wall like this. And let it reach the micropyle like so. If you want to color in your diagram, feel free to do so. So here in this diagram, what happens is the pollen tube reaches the micropyle and the pollen tube nucleus will disintegrate. So the tube nucleus is no longer here. The tip of the pollen tube will burst and discharge the 2 sperm cells into the embryo sac. So in this drawing, my sperm cells are still here. Okay. These are your sperm cells. The tube nucleus has disintegrated. So at this point, the pollen tube is basically bringing the sperm cells to the micropyle and then allowing them to go in. After this, what happens is sperm cells will go here and go here. Let us move to the third diagram. But first, let me just give you some time to draw this. And we're moving on to the third diagram. The third diagram in our series, once again, I am drawing the carpel. I'm drawing it big so it's easier for me to do my drawing. It doesn't have to be beautiful, as long as you can do the labeling and understand the content. Okay, so here's where the drawing is going to look a little bit different. I'm still drawing the pollen tube and the pollen grain. Here you will still have your synergid cells, but now you have something different inside. Now in the middle here, you will have a zygote. This is a zygote, which is the egg cell plus one sperm cell. And it will later develop to become the embryo. In the middle of this thing, you're going to have what is called an endosperm. So we're going to color it differently, and I'm going to label this as the endosperm. This endosperm is the result of a sperm plus two polar nuclei. This endosperm will later develop and become the nutrient tissue, and it's going to help the new seedling as it grows in a new environment. Okay, so that is the endosperm and the zygote. Endosperm is 3N. The zygote is 2N. And as you notice, there is one fertilization here and one fertilization here, which is why it is called double fertilization. Here is where double fertilization has occurred. And there you have it. Okay. Giving you a bit of time to have a look at this diagram. And we'll have a quick review once again. With double fertilization, we start with the pollen grain arriving on the stigma. The pollen tube will germinate, bringing the tube nucleus down with it, goes down the style and around the ovary wall. Following close behind the tube nucleus are the sperm cells. As the pollen tube reaches the micropyle, the tube nucleus will disintegrate. The tip of the pollen tube bursts and discharges two sperms into the embryo sac. And in the final stage of double fertilization, both the male gametes or sperms will fuse with nuclei in the embryo sac. One sperm will fuse with an egg cell to produce a diploid zygote. Another sperm will fuse with the two polar nuclei to produce the triploid endosperm, which is triploid. The ovule will become the seed. The integument will become the seed coat. The ovary will become the fruit. The zygote will develop into the embryo, and the triploid endosperm will undergo mitosis and later develop into the nutrient tissue. So that is all for this double fertilization parts. So this entire subtopic can be summarized in a series of diagrams. I hope that you're able to follow along with the drawings and that you would refer to your lecturer for any updates, for any parts that you don't understand. And please upload your drawings in the slots provided in your own Google Classroom. That is all from me. Thank you very much for joining in on this lesson, and I will see you again soon. Bye!